One experiment that led to the foundation of DNA's purpose was conducted by Frederick Griffith in 1928. Griffith knew that bacteria could swap a transforming factor that changed them, and that this transforming factor must be the basis of heredity. But which biomolecule was this factor? In his experiment, Griffith used two different strains of pneumonia. The first strain he used was the S strain, or the smooth strain. Now, this strain covers itself with a polysaccharide capsule, which protects itself from the host's immune system. And because the strain defends itself from the host's immune system, the host is unable to fight the bacteria and therefore dies. Now, these two strains are surrounded by red, and the red is supposed to be the capsule, which protects the strain. The second strain is the R strain, or the rough strain. And the R strain is not covered by a capsule, which allows the immune system to fight and kill the bacteria, allowing the host to live. Now here the strains they don't have a capsule, which they're not protected. Griffith injected the third mouse with heat-killed S cells. This proved that the heat-killed S cells were harmless because this mouse lived. The fourth mouse was injected with heat-killed S cells mixed with living R cells. For this injection, the bacteria from the S strain were killed off by heat and the remains were added to R strain bacteria. And this process made the harmless R strain able to kill its host. Because the DNA of the S strain survived the heating process and was taken in by the R strain, the S strain DNA contains genes that form the protective polysaccharide capsule, making the R strain having qualities that would not allow the mouse's immune system to kill off all the bacteria. Because the mouse now had the gene of the protective capsule, this mouse died. Because the remains of the S strain caused the R strain to contain qualities of the protective polysaccharide capsule, Griffith suggested that bacteria are capable of transferring genetic information through a process known as transformation. The next experiment that laid the foundation for DNA's purpose was the experiment conducted by Avery, McCarty, and MacLeod. This experiment was conducted by Oswald Avery, Colin MacLeod, and Macklin McCarty in 1944. Before their experiment, it was believed that the proteins were the carrier of genes in cells. However, their experiment led to the discovery that genes and chromosomes are made in DNA. The concept of their experiment was that the bacteria are able to transfer genetic material through a liquid form to other bacteria by transforming them. If bacteria transforms when a molecule is placed in liquid with them, then that molecule holds genetic information. This team focused on the idea of whether the transforming factor of the S strain was a protein or a nucleic acid. The three scientists attempted to transform the R strain into the S strain by incubating living R strain and heat-killed S strain. Avery, McCarty, and MacLeod extracted purified DNA, proteins, and other materials from Streptococcus pneumonia S bacteria and mixed R bacteria with it. Only those mixed with DNA were transformed into S bacteria, resulting in DNA being the transforming factor, not proteins or other materials. To conduct the experiment, the scientists treated each sample with an enzyme. The scientists used four different test tubes to conduct their experiment. Each test tube received an enzyme with a different task. Test tube number 1 received an enzyme that destroys lipids. Test tube number 2 received enzymes that destroyed carbohydrates. Test tube 3 got enzymes that destroy protein. And test tube 4 got an enzyme that destroys nucleic acid. They treated each sample with an enzyme that destroys a biomolecule. The next experiment was conducted by scientists A.D. Hershey and Martha Chase. Both scientists worked with DNA virus T2, which infects E. coli. The steps in this experiment were infection, blending, and centrifugation. Hershey and Chase discovered that when bacteriophages containing radioactive P were able to infect non-radioactive bacteria, all infected cells became radioactive, and a lot of the radioactivity was passed to later generations of bacteriophages. When the bacteria were infected with bacteriophages labeled with radioactive S, and the virus coats were removed by whirling them in a blender, almost no radioactivity could be detected in the infected cells. The DNA element of the bacteriophages is injected into bacterial cell, while the protein piece remains outside. However, it is the DNA that directs the formation of new virus particles, complete with protein coats. The next experiment was conducted by scientists Rosalind Franklin and Maurice Wilkins. Franklin and Wilkins led separate research groups and had separate projects, but they were both concerned with DNA. The two peers did not work well together at all. Franklin was given responsibility of a DNA project while Wilkins was away, and once Wilkins returned, he began to treat Franklin like a lab assistant. Wilkins even showed scientist Watson one of Franklin's crystallographic portraits of DNA, which immediately went into an article and up for grabs to the public. The scientist John Randall handed Franklin the responsibility for a DNA project which nobody had worked on for months. She also took many X-ray photographs of DNA and suspected that DNA was helical. She concluded that there were two anti-parallel sugar phosphate backbones with nitrogenous bases paired in the molecule's interior. Wilkins was skillful in isolating single fibers of DNA and gathered some data about nucleic acid structure after Franklin joined the research team. Two Cambridge University scientists, James D. Watson and Francis H. C. Crick, conducted another experiment. Their research and experiment determined the double helix structure of DNA. Watson had attended a lecture by Rosalind Franklin and used the information he acquired from the lecture to benefit him and Crick's experiment. Based on the information from the lecture, Watson and Crick made an unsuccessful model, which resulted in the head of their department telling them to stop the research on DNA. However, the two scientists did not give up. Using Franklin's information, the two scientists came up with a theory that DNA was made up of two chains of nucleotides, each in a helix, with one going up and the other going down. The matching base pairs interlocked in the middle of the double helix, which kept distance between the chain constant. They proved that each strand of the DNA molecule was a model for the other because during cell division, the two strands separate and on each strand, a new half is created. This allows DNA to reproduce itself without changing its structure. The next experiment was conducted by scientists Matthew Meselson and Franklin Stahl. They designed an experiment to determine how DNA replicates, and it tested the hypothesis of DNA replication. Meselson and Stahl gave evidence that each strand of DNA served as a model for new synthesis called semi-conservative replication. In their experiment, bacteria cultured in medium containing nitrogen-15 was transferred to medium containing nitrogen-14. After about 20 minutes, the DNA sample centrifuged into the first replication. After about 40 minutes, the DNA sample centrifuged into the second replication. The results of this experiment show that after one generation of the double-stranded DNA is one half heavy from the parent and the other half lighter, the one newly synthesized. The DNA strands serve as models for their own replication. Thanks to the research and experiments of the scientists we just discussed, we now know the foundation for DNA's purpose.